Hey, Kevin.
Hi, Alexei.
So let's see if more people show up.
Mm. Well, it's a Sunday evening in Europe, so I
Yeah, I
hope.
think it was a bad idea. We can, like, what we can do, we can release the recording and do another Q&A in, some other more convenient time. But we can wait for a couple more minutes and
Mm.
see if more people show up.
And I was thinking maybe uh, once people have read into the challenge, they understand what they need to do, right? They focus on the challenge itself. Then we would tell them, hey, instead of rebuilding the wheel, you can already use a framework and use Omics-OS for that. As I said, like I truly believe that this would just make it faster for them to actually go to the science.
Yeah, but I know that many people already chosen the challenges. So
Okay.
like, and for instance, Dmitry he already built his first prototype. So like he was the fastest gun on the in the West. So, yeah, so it's a good time already. But like we can redo it later. So I don't. Yeah.
Hmm. Okay.
Okay, we see we have more people finally. Yeah, so it's a good time already, but like we can redo it later, so don't.
Yeah, no, don't worry. I mean, we can
Okay.
also skip this one. Thing makes more sense if people are a bit more prepared and stuff. Um, but maybe I can give just a super quick intro here.
Yeah, let's
Um,
wait for more people. The thing before it was a lack, so it basically repeat what I said twice. Let's wait for until like 2-3 more minutes.
sure.
Hello, Ryan. Do you have a team, by the way? Are you working on something in challenge?
Yeah, our team's doing the Retro Biosciences one, but still was interested to hear what Kevin's up to. And
Stanislav, do you have a team and challenge to work on? Stanislav, can you hear us? Anyway, I think I don't believe many more people will join us. So let's start from this small group, publish it, and see what happens. I will also. I was also asked to publish. We have a Telegram and WhatsApp groups for those who don't know how Discord works or don't like Discord. So we set up these groups as well for everyone. Okay, so Kevin, the floor is yours. Welcome.
Well, thank you so much. Hello, everybody. Big group, but I think this is going to be a short talk. So, first of all, I think it's amazing that you joined the hackathon. This is This is one of the more important ones, especially considering this is 20 days and a bunch of smart people from around the world are contributing to it. So the reason why I partnered with the hack is because in the last 2 months, I built Omics-OS just because of the reason that the last 2 years, I was working with omics data, proteomics, and genomics. And I was working with the largest biobank, the UK Biobank, and you have no idea how much time we spent into handling this data, from cleaning, pre-processing, filtering, QC, all this kind of stuff. So I was thinking maybe this is definitely something that should be automated, especially with AI, as it takes away time from the actual science. And when we talk about aging, instead of reinventing the wheel, we should focus on actually finding out what is causing it, how to reverse it, how can we be faster with the tools that we have. So I'm going to quickly give you an overview of Omics-OS, just quickly show you how to install it and how to run it and what it actually is. Do you see my screen?
Yes.
Perfect. So the goal of the first product called Lobster, is to replace bioinformatic tasks with natural language. And it's not a ChatGPT wrapper. It's a framework that allows you to do those things reproducible, loggable, and iteratively. So the best thing would be that the packages that we normally use in bioinformatics for all kinds of pre-processing steps are already part of it. And I mean, most of them are open source, which is pushing the frontier of science. Now just made in an automated way. So agentic systems using them for different tasks, for different problems. Maybe as a quick overview. So currently the architecture, I'm just going to show you a very simple version of it. The architecture is rather simple. I mean, still took 2 months, but it's rather simple. It's a supervisor multi-agent system built on Python and LangGraph, which are built on top of tools, services, and adapters. This architecture is really important because it allows you to plug and play different modalities into the system without having to reinvent the wheel. In the center of it all, there is a data manager, which allows you for context engineering, so that your different agents always have the same context and can extract parts of it depending on their needs. I won't go too much into detail because you can check everything for yourself on the wiki. But what is important here is to know that the system is built to be flexible for new problems, for new challenges. So those adapters are made to have new data types, services are made for new tasks, and tools are made to use the adapters and the services, making it accessible for the agents. Currently, we have 5 to 6 agents without a supervisor, and I don't know which problem or which challenge you would choose, but I will show you quickly a demonstration of a few of them. I think one of the more interesting ones is the research agent, which is connected with the NCBI API system. So the Entree API system, meaning you can not only search for publications, but all the data sets that are connected with those publications. So I'll show you in a second. This is really speeding up the process of finding a data set that might help you in answering a question for your challenge. Maybe when it comes to female health, right? Female reproductive health. And you cannot directly look for a data set with only females, which is super hard to find, but you can find data sets which include females in the cohort. And this is, for example, given in the abstract or in the meta files. So the research agent is still buggy. It's still rough because of non-standardized data uploads, especially when it comes to transcriptomics. But it really helps to iteratively search for this kind of stuff. Then the data expert is set with tools to handle the download, the extraction, and the preparation, like loading this kind of data into the system so it's accessible. And right now, the only kind of data supported is transcriptomics. I do have proteomics already in the pipeline, but I want to make sure that it's bug-free or that it's at least workable. So throughout the next couple of weeks, this will be accessible as well. The ML engineer or the ML expert right now is more for niche tasks. For example, once you research your data set, you downloaded it, maybe you made some clustering, so prepared or checked for quality, and then you want to export it into machine learning ready formats. You can use the machine learning expert. For example, you want to train foundation models or a classifier or whatever. And then, of course, a transcriptomic expert, which is split into single cell and bulk. I don't know if you've worked with transcriptomics before, but right now, this is probably one of the most important fields generally in research, just because it's cheaper than proteomics. You can look deeper. Single cell is more granular than bulk, but it's also more expensive. So it's currently the best level that we have, or the most adopted level that we have in research to understand also mechanisms. And the visualization just uses some visualization tools for you to have an output then. So long term, right now, I'm working on the left side, which for you might be relevant for especially the knowledge graphs, as they contain knowledge. They contain publicated knowledge, confirmed knowledge, without having to hallucinate. It's like a RAG model. For example, if you're focusing on marker genes in one of the challenges was bladder cancer. Let's imagine bladder cancer, right? You need marker genes, then you don't want to just generate them. You want to get them from a knowledge base, and this would be done by knowledge graphs. And harmonization, I don't think this is going to be a problem right now for you guys. This is more of an industry problem, but yeah. So if you want to just, let me just quickly show you how to use it. You go to Omics-OS, you press on the download, and you end up on the GitHub repository. So from here, just follow the README. It's rather simple. Copy it. You clone the repository. Make sure that you have UV installed. It speeds up the process quite a bit. UV is like a pip package manager, really fast, built with Rust. I can really recommend it. Okay, once we've installed it, we go to Lobster local and check the README. What you do need here is an API key, because it uses Claude, the Claude API right now. So you can either get your own Anthropic API key, or you can text me if you want to use it. I can give it to you, so you can test it a bit and see if it's useful or not. Also, for the extent of the hackathon, I provide free API keys if you're using Lobster for your project. So if you have a Mac, beautiful. If you have a Windows, I'm so sorry, I have not yet tested it on Windows. But for Mac and Unix environments like Ubuntu, easiest is just make install. I would suggest you use make install global, as then you could use Lobster in another environment where you have, for example, other data. But it doesn't really matter. You can also just make install it. I should have set it up so that you don't have any problems with the environment, but you never know. It's always working on my machine, but let's see. I started 2 months ago and launched last week, so there will be bugs, but I will actively develop it during the hackathon, so these can be fixed in a matter of hours and not weeks. So while this is validating, let's check. Okay, we have the make install. We open the end file where we add our activation keys. Yeah, Ryan, you have a question?
Yeah, is the project description up on the hackaging.ai website?
I think there is a link to Omics-OS there and nothing else in partners. So you can go to Omics-OS from there and yeah.
Okay.
Good question.
Yeah, go like to the partners.
Oh yeah, here you go.
Omics-OS. You try to click it. Yeah.
Perfect.
So.
Nice.
So then what is the project? Just hack on this. So you've started like a scaffold of an AI agent orchestration to do bioinformatics and what's the 2 week or the deliverable project that you were thinking, or is it just kind of free form?
No, this is not a challenge. This is for you guys to not having to reinvent the wheel in bioinformatics without having to use super complex academic repositories.
I
So it's
see.
really to make you faster and to have a starting point. There's quite an extensive documentation which you run Claude and create an init file, and then you can directly interact with it, vibe code it or vibe extend it.
Thanks for clarifying.
Sure. All right, so now that it's validated, we activate the environment. We open the end file. And here you can see that there's two options. Either you add the Anthropic API key or you add the AWS Bedrock API key, right? So the problem is with Anthropic, for some reason, they changed the quota, meaning there's like a limitation in the requests you can send, and especially in agentic systems where it's like iteratively, you come to this limitation quite fast. That's why I suggest you reach out. I'll give you an AWS Bedrock key, which really makes it easier. So I'm gonna use mine here.
Sorry, what is the best way to reach out to you? Can you write it in chat or
Yeah,
say as well?
of course. Reach out via email. It's kevin.yar@omics-os.com. If you wait, where is the chat? Here we go. I'll be developing every single day, so I'm ready for all your problems. Yeah, just reach out. All right, we added the environment key and we are good to go. Lobster should be ready now, and I really hope that there is no bugs because I did some pushes yesterday. Oh yeah, we call it with Lobster chat. I highly recommend you to add the reasoning flag because this shows you the thought process of each agent. There's a quick menu. Those commands are important for you to interact with your current system. So Lobster is running on is living in your folder. It's just like Claude-Code. You can access files, you can read files, you can modify files. Exactly. So you can see those different agents have been loaded. And does one of you have a specific topic in mind that you want to search data sets for?
How about inflammaging?
Inflammation?
Yeah, inflammation, sure.
Or did you say aging? Sorry, I didn't hear you.
Well, there's inflammaging, but that's kind of a buzzword. I guess you could
Ah,
pick inflammation or something like that.
well, let's maybe search for inflammation of lungs. Okay, then search me data sets in, and for in research, for example, in aging research, we first want to understand, okay, which proteins are behaving differently in maybe in older people compared to younger people, right? So let's search for data sets of of lung inflammation using single cell transcriptomics in human from the last 10 years, as we don't want to use the older transcriptomic analysis. I need at least 20 samples. Now let's start with this. All right. So the research agent is now, first of all, creating the initial query. Sorry, first the task is being given to the research agent, and then it does the iterative research. I'm not sure if the current way is the best way. As you can see, it just uses smart querying strategies. No embedding, search, nothing. It just uses the standard API system. But I do think it does save you some time, especially as it can also peek into the metadata and get more information about the samples. The cool thing is you can run this in parallel. So if you're searching for some data sets here, you can already load your data set somewhere else. I make an example. I have, let's get out of here. Documents. So I have, okay, data is gone. If you have data, if you have a set of data somewhere lying around, either in CSV files, in FASTQ files, in H5AD file, and you want to read them, the adapter architecture makes it very easy to be flexible with what kind of data comes in. So you just add a read command, and then you just regex expression like asterisk H5AD. But yeah, this is all described more in depth in the wiki. And as I said, reach out if you have questions there. All right. I think I just let you ask some questions if you have any. And yeah, go ahead.
I have a question. How to search hypotheses about synergies of aging treatments in data sets, anti-aging interventions? How to search hypotheses in data sets? How you think it can be possible? Not in just articles.
Do I understand you correctly that you want to search a hypothesis inside a publication?
No, I think the question was about like if we can search data sets where we can test this and this hypothesis, for instance. Right, Dmitry?
Yes.
So first you start with your hypothesis, of course. Let's imagine your hypothesis is creatine slows down aging. I don't know, something like that. So then you're starting with, all right, let's maybe see how creatine affects the body. Or maybe let's first of all check publication. First of all, I would do some literature search, right? I personally use Consensus. I'm gonna. This is a search tool for publications. It uses vector searches, so it's really good. There's others one as well, but yeah, just use it. Then you do a literature search. You see, okay, is there any connection? Is this something that I could dig in deeper? If yes, and you have your maybe you have your target proteins that are affected by creatine, or you know maybe which pathway, or not even that. Maybe you just know, okay, creatine has an effect in your body. Then you start with searching data sets where people have taken creatine and control groups. So people with creatine and control groups. Preferably, if there is a transcriptomics layer, you search for this. So once you have your data set, or the search itself would be find me data sets with a cohort of creatine and a control group with this sample size and so on. So is this answering your question?
Let's start. Yes.
Okay. Any other questions?
Yes, I have already another question. How you said that I can connect some plugins maybe to your system.
Mm-hmm. What plugins are you thinking about?
Yeah, yes, and I have some plugin about how to get cost of wet lab consumables to test hypothesis. So I need for this hypothesis price of what it will be to test. How I can connect to search of wet lab components, consumables to your system, to search all at once.
Do I understand correctly, you already have such a tool and you want to connect it with Lobster, or do you want Lobster to search the prices of those wet lab components?
How you will do it for your system? In your system idea of your system, how what I need? I need to make this as some task for your already agents which already have. I don't see which agent can do it in your system, or I need to make my own system, my own agents for your system. How you think it will work?
If it's a one-time task, I wouldn't recommend you to use Lobster, just use ChatGPT. If it's something that you want to offer as, like a system where people have a question and then you get the wet lab costs, then I would recommend you to create an agent, which is described in the wiki. And based on this agent, you create the right tools. And then you can just run your agent with a web search, for example, right? Then you would need web searches to go for that. This is quite non-biology. It's a lot of just checking the internet. So I'm not sure if Lobster would be the best solution here.
No, it's not one time. It's about 10,000 hypotheses to test. And for them, I need consumables to be filled automatically.
Okay. Yeah, maybe you can send me a bit more information via email. I can look into it and help you finding a way or think about how this could be useful.
Yeah, thank you.
Of course. Ryan?
Yeah, wonderful work, Kevin. Like, I think you're pushing in the right direction. I guess so. Sounds super cool. I want to forward it to my team. I'm coming to this from as a software guy. I don't know all the biology. But maybe just wondering, like, how do you see this product comparing to some other things? I know there are similar and if this is off base, you don't even have to answer, but I know there's things like Science Machine and there's a Future House, for example. Like, how are you thinking this is different, and like what's the niche that this is getting maybe that those other guys aren't?
So first of all, it's open core. You see exactly how it works and you can customize it the way you want. And
Perfect.
second of all, I think this is really focusing on local development as well. Because, if you're working in biotech, if you're working with sensitive data, you don't want to share it with just a company out there, startup especially. So just industrial wise, it is quite an edge to have something local, running local. And coolest thing would be if you even have a local LLM doing everything local. It might not sound very useful for you right now, but I've worked in pharma research, and sharing sensitive data it's quite tough.
Yeah, there's still a
And
lot of vertically integrated companies. I get
Yeah,
that.
I also can confirm it working with pharma all of my career.
Sweet, thanks.
sure.
Gentlemen,
All right.
any other questions?
Yeah, so thank you, Kevin, for this great presentation. So I just quickly checked your website, and so far I could not see is it capable of doing things like quantitative trait locus analysis, or it's like still planning it to implement in the future.
Can you elaborate a little bit?
Yeah, so yeah, for example, to assess how like for example, given genetic markers, they correlate with the corresponding quantifiable phenotypes. So like to do the scoring, like to evaluate that's like really quantitatively the connections.
No, that's a good point. So I started with transcriptomics as it was the most important one right now for industry and for my customers. The next one will be genetics, but there's a bunch of different challenges that comes with it because there are, old repositories that are widely used in genomics, and installing this on your local system without Docker, it can be tough. Not everybody knows how to use Docker, so there's a choice of how to set things up. But to answer your question, if you have a specific request, I can look into it. As I said, like genomics would be the next step, and we can definitely check this out.
Okay, then I can check. I can send you the more details about this technical task via email later.
Sure, do that.
Okay, I don't believe we did have any other questions. So last chance to ask Kevin on this call. Otherwise, let's thank Kevin, and also for those who listen it in the recording, also reach out to Kevin via the email. We'll probably add it to the description or the first comment. And you also, basically, Kevin mentioned this email in the video. I think Discord also. Are you in Discord, Kevin, in the Hackathon Discord?
Yeah, I'm trying to be active. If I don't answer immediately, just send an email. I'll always be there.
Yeah. Okay. Thanks a lot. Thanks a lot for showing up in the Sunday morning or evening, depending on where in the world you are. Yeah.
Sure. Thanks for having me.
Let's hack aging.
Okay.
Sure.
All right. Hear from you soon.
Bye.
Bye bye.